Welcome everyone to the IBSS Seminar Series. Big shout out to Alora. If you don't know her, raise your hand. She's organizing this, great job. And Kel, and also today I'm really happy that I get a chance to introduce the speakers on this incredible team that's addressing, I think, not just a fundamental biodiversity issue for California, but also I think it's going to elevate entomology and CCG, our genomics facility here at the Academy. Now, as some context, we all know that biodiversity loss is a major issue across the planet, even in California. But I think most people don't really appreciate that for most biodiversity, we don't have any data. And we can refer to this group of taxa that lack data as the dark taxa. And for those who don't know, the home of dark taxa is entomology. And by teaming up between this initiative funded by the state of California and the CCG, I think we have an incredible opportunity. First, this initiative is really developing the really first protocols that will leverage a museum-based data store that we have here. But at the same time, the advantages to entomology as a field, as a collection, I think are threefold. One, it actually allows us to address this taxonomic impediment, that's the dark taxa. Two, it addresses that we don't have a baseline data to actually understand change in the planet, like in California. And third, I think it really allows us for the first time to think about taxonomy as a applied solution, that we can actually, in a short time period, begin to use our taxonomic skill sets to actually solve problems. So without further ado, and I welcome you all to give a round of applause for this incredible team, starting off with Chris Grinter. Thank you, Chris.
Thanks, Brian. Good morning, everyone. So yeah, I'm going to dive into this exciting update that we have from entomology. We've been busy for the last 18 months or so, working hard down in the basement, and I'm going to start by introducing our team. I'm going to set a timer here so I don't go completely crazy with my... We have a few speakers today, so I'll be followed up by Julia and then Anna, who's going to really dive into all of the details and methods of this project. But I'm Chris Grinter, Collection Manager of Entomology, and on the curatorial team downstairs is Julie and Diana. You'll hear from Julie in a second. We have an excellent team of hardworking interns, Kate, Sarah, and Levi, who've been with us for several months now, chugging along through every aspect of this project, from collecting insects to getting them ready to go into the molecular lab. Then we have our amazing genomics core team, helmed by Athena, with Anna, Holly, Cece, and Rowan, and Anna will have an ample opportunity to introduce you to more of what they're doing. So the project really comes from a foundation in 2018, 2019, when we start seeing a lot of reports of the insect apocalypse. And in 2019, at the Entomological Society of America, there was a symposium that was organized by Dave Wagner and a few other people that hosted a bunch of symposium speakers talking about kind of the shocking data that was coming out of mostly Northern Europe about insect decline and what was being branded as the insect apocalypse. It certainly made a lot of headlines across New York Times and all sorts of media outlets. But this question was really circulating and percolating to this paper, which came out in 2021, that Dave Wagner wrote with a few other people, The Insect Decline in the Anthropocene, Death by 1,000 Cuts, which summarized nicely about a dozen papers that were looking at impacts of climate change, agriculture, invasive species, all these things that were impacting biodiversity on a massive scale that people weren't really paying attention to. I think all of us can anecdotally look back and say, our windshields are cleaner than they used to be from, 10 years ago or 20 years ago. But it's just anecdotes. We don't have the data to really back that up and say, look at the numbers of insects over time. They had been doing this in Northern Europe pretty extensively, so we have a great data set where we can say pretty definitively, look at the biodiversity over time. But in North America and the rest of the world, we haven't been doing that. There's a few great data sets out of California. Art Shapiro has amazing butterfly observation data sets from the Sierras down into the Central Valley that really shows staggering loss of butterfly populations throughout California. But we need a lot more than that. There needs to be a lot more that's happening than just some local butterfly observations or honeybee observations or things that are really, really different than what we need to be doing. So in February 2021, this guy, Dan Glusenkamp, reached out to a whole bunch of entomologists. He wasn't an entomologist or part of our community, so we didn't really know where he was coming from. And he was best known at the time for discovering the Franciscan manzanita, this little shrubby manzanita here in San Francisco that was thought to be extinct. But he had some really recent success getting money from the state to seed bank California rare plants. And based off of that success, and now pivoting as a role at the California Institute for Biodiversity, Dan was looking to expand the horizons of what he could do and money he could get from the state of California to really impact other research around the state. And so he came to us and we started putting together a consortium of California entomologists, basically. We met on Zoom all the time from all these institutions, had some rather large Zoom calls over and over again, brainstorming what sort of projects could we do if we had to write ourselves a blank check, like what would be impactful for the state and how would we divvy it up and how would we all participate in something like this. And the CIB would be our lobbying wing, essentially. Dan would go to Sacramento and get the money from state legislatures so they can put in a direct member request into the state budget, which was perfect because in 2021, California had a $46 billion surplus that they were looking to reinvest into California. So there was money to be had. Just a fraction of that 46 billion would get us up and running for a very long time. But we did a lot of organizing and meeting, and we finally decided to meet in person. And Dan's connections through Sacramento, he said, well, Governor Brown has offered us his ranch in Colusa. Why don't we just go and camp over the weekend and do some collecting and start thinking about what we could do? Well, we went up there and didn't expect to actually spend the weekend with Governor Brown. He showed us his ranch and let us swim in his pool and drink his beers and talk biodiversity, which was a really fun experience. And, it was a little unexpected and really exciting to get him, who's been talking about biodiversity his entire career, to look at a trap sample of insects and say, like, oh my God, this is biodiversity? Like, there's thousands of insects flying in his yard in Colusa County. And it just kind of blew his mind and opened him up to understanding what we're talking about with biodiversity in California. And we also got to meet his family and dogs. This is Colusa, who does not want to go on a walk in a toasty afternoon in Colusa County. And so we started congealing a plan of what to do around DNA barcoding, and 4 main project goals kind of solidified. And it was to collect new specimens from priority locations across California to help fill in the gaps in places that we don't know very well. And we're going to barcode and secure our existing collections. So we're going to dive into the collections here at the Academy and utilize those to generate sequences for this public database. And we're going to expand opportunities through development and training of people here in California. And we're going to deliver all of this freely to the people of California pursuant to these California biodiversity orders, executive orders, and now, of course, California 30 by 30. And so why are we going to do DNA barcode? It's a very, you've all heard it. Maybe not everyone has participated in barcoding, but it is a very simple, affordable technique to get a standardized DNA region out of the mitochondrial genome to help identify a species. And it really can democratize species identification. You don't need to be an entomologist or an expert to take $10, $20, send it to Canada, have the Guelph Center for Barcoding sequence it for you, and you can get an identification back. Of course, that's based on the reference library that they're pinging with those sequences to see if they can actually match it to a known animal. That's where the complexity comes in and where we can really fill the gap here and say, we have one of the greatest biodiversity hotspots on the planet. We should be contributing to this knowledge base as much as we possibly can. And it really modernizes our specimen collecting techniques. Entomology can be a very Victorian sort of feel where you're out there with lanterns at night collecting bugs, but we can also do this value additive sequencing to really unlock their potential for generations. I mean, the amount of material we're generating is going to be keeping us busy for a very long time. But when you Google what is DNA barcoding, you see this graphic that shows up everywhere. It's very misleading because it says you take a species And then you have DNA you take out of it and you get a barcode that matches that species. Well, that's just, it's not really true because that's meaningless without taxonomists and collections in the beginning. All of those species IDs come from a collection in a publicly vouchered, repeatable way where there's an expert in the field that has authoritatively identified an animal. From that, we can take the DNA and make a barcode to match it back to that animal. And without that, it's just meaningless DNA sequences. So really, we're building a bridge between a collection like this and applied sciences. And it's a tool, a deliverable from this project that will be widely available to anyone who wants to use it. And we're really going to be establishing this baseline for biodiversity monitoring in California. And these data sets are going to be useful for generations to come. We're really creating a lot of material, which is going to be exciting. So the question was, how many insects are there even in the state of California? We really don't know. That's a great question because we've been at it here at the Academy for 170 years, and we have about 25,000 species of California insects. No one expects the Academy to have all of the insects of California, let alone maybe 50%. I would be shocked if we had 50%. So I think reasonably we could say 40, 50,000 species in the state, but if we got 100,000, I don't think anyone would be shocked at that number either. So as part of this, we're actually just going to be building a list of California insects and then sequencing as much as we possibly can. And what do we even do with 100,000? How do we manage that many specimens? Do we outsource it to Canada, or do we build capacity in state? And of course, this is a state-funded effort. We build capacity in state as best we can, and we're going to utilize some sequencing in Canada just because it's cheap, reliable, and easy. But we're going to be spending, and you'll hear all about how we have invested capacity in the state with developing really exciting new techniques to do this sort of stuff. So in May of 2022, the California Insect Barcoding Initiative was established with our first tranche of funding for phase 1 of the project, which was new collections. So getting out there, collecting as much as we possibly could, hitting the ground running with Diana and Julia right away. This is us in the Eastern Sierra. We met up with Dave Wagner and some other colleagues and had an amazing trip, which included going down to the Mojave, and we off-road tested our Toyota minivan, which did not end very well, surprisingly. But we got out of there pretty easily and had a wonderful trip. So we were all over California this last spring setting up these observatory sites. And basically, it's a standard insect collecting trap. This is a malaise trap, which is basically just a tent where flying insects and occasionally some crawling ones just go into it, get funneled into a bottle, and that bottle is collected every single week by either us or a project volunteer who then stores it in the freezer and waits for us to pick it up and sort it, barcode it, everything else. This one on the left here is from Siskiyou County and is in Glenn Fine's yard, and it comes with a guard dog, which is great because he's had a bear tear that trap down at least once before. But it's producing an amazing amount of data. So we at the Academy have 6 of these throughout the states, and project-wide we have about 40. And some people are only managing to collect them once a month, but we're doing it every single week for at least a year on all of our trap sites. And then we're also going to be weighing some of the biodiversity that comes out of them so we can apply some other questions to these, like how much is actually flying at a given time of year. And this dovetails beautifully into an existing project that's completely separate, which is Project Life Plan, based out of the University of Helsinki. And Brian's a PI on on that project. But it's a global effort to establish these baseline biodiversity collecting standards where we have a standard malaise trap, and for Life Plan, there's also spore sampling, camera trapping, all this other stuff that will be part of this long 6-year effort. And so we're able to cooperate a lot with Life Plan. We're going to share data. It's going to be an exciting project, and this one's on our roof here, so I'm sure you've seen it by now. If not, go take a look because it's going to be moving back to the Presidio in just a few weeks after the start of the new year. And so with that, I'm going to hand this over to Julia, who's going to talk to you about the boots on the ground effort that we've had over the last 18 months or so, exploring California. So thank you very much.
Thank you, Chris. So as far as new collections go, I guess I'll reintroduce myself. I'm Julia. I'm mainly working on the new collections, and I started on this project a little over a year ago. So as far as new collections go, these are all of the different places where we've collected over just a little over a year in California. So those malaise traps that you just saw on that last page, the observatory sites that we're collecting once a week, those are in purple. There's actually two in one of the spots because they're kind of close together. They're in the East Bay, and we're actually collecting those. And those, like Chris said, are collected once every week. All of these other areas are snapshots, so they're times that we've done other forms of collection. And why do we need this over the entire state? So as Chris said, California is a biodiversity hotspot, which means there's a lot of different ecosystems, which leads to a lot of different flora, which leads to a lot of different fauna, and those fauna include insects. So we need to get every possible place we can in the short amount of time we have. So just some fun statistics over the past little over a year. We've traveled around 7,500 miles, collected tens of thousands of specimens already, those collection sites range from 0 to 8,500 feet of elevation. And we've worked really closely with 9 UC reserves, as well as obviously we've collected many other places as well. So this is just an example of all these different ecosystems. All of these pictures are places that I took photos of in just different locations, collection locations. So you can see there's so many different ecosystems, and we ideally have to catch every insect in all of them. So once we get to those locations, how do we collect them? So there's many, many different forms of ways to collect. These are a list of many that we've used, as well as some other ones. So here you can see us doing different forms of collection. This is Diana setting up one of the malaise traps in the East Bay and me collecting. So these ones we are personally collecting every week. And then this is a UV trap. So we'll set these out at night, and the light will come on and it'll attract a lot of insects. And so sometimes when we set this out in the morning, when we go get it, it looks like that. So that is literally inches of specimens in a bucket. So obviously that's a lot to go through, but we don't need 100 of the same thing. And this was an emergence event, so it was a lot of the same thing. So when we get this is what the day looks like for us. It's a lot of sorting and a lot of trying to figure out what we need. So we ideally want 1 or 2 of everything that we collect in each site, because even if you have the same species at different sites, they could have slightly different genetics as well, because they're geographically isolated. So here you can see us sorting, and then we start to pin our specimens or put them in alcohol, depending on what we're catching. When they're finished being pinned, they look a little bit like this out in the field. And this is another example. Sometimes we also have some of these really small organisms in this like little box we've got over here, and sometimes we will spread some of our leps, so our moths and butterflies. So once we get back to the academy, we have to prepare all of them further for the collection. So all of our specimens, other than the ones in the alcohol, are going to go into a dehydrator. This is the same thing that you would put food in to dehydrate, and it will lock all of their limbs in place, including these moths. So here you can see that they were spread, and then they get moved off once they are dehydrated. Some other examples are sorting our alcohol specimens. So we're able to sort all of our other specimens, but when we get back, it's easier to sort our alcohol specimens. And this is what they look like once they've all been sorted, ideally to species, but we'll talk a little bit about that later. And that would be ready to go into the collection. And then some of our small stuff, that is either going to be double mounted, like this top picture, or pointed, like the bottom picture. We also have our collection labels. So out in the field, you're not going to write every single thing down. So as you can see, the one on the left is our collection label, and then we need all of these labels to actually incorporate it into our collection. So the top one is going to say all of the different information that you need, including the collectors, the date, the type of trap, elevation, and the place. And then ideally, again, you want to identify it down to species. So we have a species determination label. And then probably most importantly, we have the catalog number, this one with the little QR code. So this is put on all of our specimens now. That QR code, when put into our database, will connect all of this information to that one specific specimen. And this will be incredibly important down the line when we're looking at DNA, because we have each individual will have its own number. So this is what they look like before they go into the collection. This would be ready for the collection. However, we're not done yet. So then we need to plate our specimens, and I might talk about this a little bit later as well. But most of what we're looking for, depending on the organism, we're either going to take a leg from the organism, we're going to take an abdomen, usually that's from like small moths, or use the entire specimen and soak it. And we have to do a lot of sorting again before plating. So we need to sort them based off of their size and then their sclerotization, or how hard the specimens are. So if you have a larger specimen, it's going to take longer in a soak to get the DNA out, and the same thing with a harder specimen. So all of these things need to be taken into consideration. And I wanted to shout out our amazing interns. Here's another picture of them. They have helped out a lot with the plating, especially in this project. So they've been a huge help, because it's hard for us to keep up with the needs of the CCG team. And then I just wanted to mention on some challenges. We've kind of touched on a few of them, but something that's really difficult for us is getting everything ID'd in time to actually be able to get the DNA sequences. So Diana and I are mainly generalists as far as entomologists go. We can ID most things down to order and to family, and sometimes genus, but genus and species can sometimes be very difficult with insects. So you can see this large amount of trays over here with one fly species, and what you probably can't see is they all say undetermined. So these ones actually we haven't collected. These have been sitting in the collection, our old collections, for a while, and it goes to show that it's very difficult to ID some of these species. Some of them are what's called cryptic, so they can look almost identical but be a different species. So a lot of the time you need a specialist in order to ID it, meaning for this you would need a dipterist or someone that specifically studies flies, and maybe even a dipterist that specifically studies this family of flies, because there are so many. So that's been a big challenge, as well as just the idea of collecting every insect in California is kind of daunting. You have to collect in every ecosystem, all of the seasons, during the daytime, nighttime, different things fly at different times of night. Everything's attracted to different things, and we're looking for a holistic approach. We want to catch everything. So that's another reason why we're using all of those different trapping methods, because, a moth might be attracted to light, but something else might easily fly into a passive trap, like our observatory malaise traps. And then some are incredibly tiny. We'll also mention that a little bit later. That's kind of a future focus, some of these tiny soil organisms. And we just don't know how many we're looking for. So when do we know when we're done? We don't. So now I'm going to hand it over to Anna on the CCG team.
Thanks, Julia. Hello, everyone. Yes, my name is Anna, and I am the postdoc for the natural history collection side of this project. So I am working at the CCG, the Center for Comparative Genomics, and I'm working with a team of 2 technicians who are in the back there, as well as a volunteer. And so we started in June. So we started later than all the other folks. So Holly and I started mid-June, and then we had our second lab technician, Cece, start in July. So we've been really focusing on getting the molecular side of this up and running for about really 4 months now. As Chris discussed, CO1 barcoding is really fantastic because it provides an opportunity for really anyone who wants to work with insects to identify something. We can go and sequence it for the specific region, CO1, the mitochondrial region of the mitochondrial genome, and with the database, given a database, someone is able to identify what that specimen is. Very cool now are approaches called metabarcoding that have really come about in the past 10, 15 years that give us the ability to take whole community samples, similar to what Julia showed you with these traps that can get hundreds to thousands of specimens at one time. What we can now do, thankfully to next generation sequencing platforms, we can take this big bulk pool of insects, extract DNA, sequence that for a CO1 barcode, and actually get a whole community perspective on what that group of insects or arthropods looks like. So what does that mean? It means that we have this really novel approach now that allows us to monitor biodiversity in a way that really hasn't before been possible. Because things are so hard to identify and take so long to identify, previously, if you had a trap like that, sorting through it and identifying everything could take you years at a time. And so now we have methods that allow us to just take one sample, sequence it, and within a matter of months, be able to know what is there. So a little bit of self-promotion, but these are 3 projects that I've done that have used metabarcoding to look at arthropods across different disturbances and experience different pressures of global change. So we could take something like leaf litter communities across a deforestation gradient and look at how the communities are restructuring given deforestation. So I worked in lowland agroforestry and cacao agroforests and looked at how leaf litter communities changed across an elevation gradient. A little bit closer to home, we can think about wildfire or other of these changing disturbance regimes and see how those affect arthropod communities. So in this case, we looked at burned and unburned sites, and we're interested to see how pollinator communities were changing. And so we were able to take these large collections of pollinators and just sequence them, and that gave us a feel for how these were shifting given changing wildfire regimes. Lastly, there's some other really cool approaches where we can actually take gut content samples or fecal matter and give us an idea of what things are eating and how that could change. So we did a project looking at endemic Hawaii spiders and seeing how invasion by this plant, Kahali ginger, here you can see it just completely dominates the understory. And we were interested to see how biotic interactions changed. And we were able to do this by looking, actually sequencing the gut content of these spiders across this gradient. So these are just some really examples of how we can use metabarcoding to understand how biodiversity is changing in the face of global change. But as we mentioned earlier, this really requires a reference library. So you get this data, you have a ton of sequences, you need to be able to know what the identity is, and that requires a database. So barcoding gives us this really cool tool, but it requires that database. Without that, you might be able to explore patterns of richness or community composition, but needing species ID really gives us this ability to just look at patterns of ecological turnover, for instance, or differences in functional diversity. If you just have class flies, you don't have much insight onto what's changing or how they're responding to different threats. So this is an example of one of my studies where we were trying to see how many individuals we could actually identify. And so of the 405 sequences we got, only 119 of them we could get to a species. So using BLAST, we got 19% of all those sequences down to a species. And that was mostly for flies. So just as an example of how little we can actually identify in these samples. And so this was for California, just an example that even in a place like California where there is more research going on, we still just don't have very much information. So Chris mentioned BOLD. This is kind of the largest database right now, Barcode of Life database, to start trying to generate this database of CO1 information so that the people can use this to start conducting these studies. So it's an incredible resource, and it's really allowing folks to put data available to the public. But there are, unfortunately, some issues with this database. There's a really large private data problem. So actually for insects, 50% of all records on BOLD are private. So if you had CO1 sequence data, you wanted to know what that was, you looked on BOLD to see what it was, there's a chance that that sequence does exist, but it's private. That's even true at the species level. So of all the species records they have, 30% of those are also private. So that really prevents it being this democratized tool for people to use when a huge amount of the data is private. It's also very biased in location. So Most of the insect records are from Costa Rica and Canada, Canada being where Guelph, the Center for Biodiversity Genomics, is based. And so it's in California and in the US as a whole, we don't have as many records as we'd like. And so it really limits us in terms of exploring these patterns. It also, most of the specimens are also deposited at the Center for Biodiversity Genomics in Canada, rather than at museums like Cal Academy. And lastly, there's a really large taxonomic bias in these databases. So insects are incredibly diverse, 27 orders of insects, but 80% of the records on BOLD are all from 3 orders, and half of those are all from flies. So we're talking about this incredible diversity, all this stuff we get in traps, so we want to know what it is, but really all we're going to be able to know what it is there is probably flies. And that was the case for our study as well. Almost most of what we were able to identify as species was from flies. And so we want at CAS to utilize this incredible collection we have to start developing a database that's going to be publicly available. All the data from it will be able to be utilized by anyone who wants to use it. And as both Chris and Julie mentioned, the power here is that we have IDs associated with every insect. We know what they are. And so when we at the CCG generate the sequencing data, we can tie that back to the catalog number and know exactly what that insect is. So we are going to generate this massive amount of data with associated specimens, so vouchered in the museum, as well as species IDs. So this is what really is the powerful part about the project we're conducting. So from our side, we're balancing these 3 big things. So of course, we want to generate CO1 barcodes, so effectiveness. How do we do this in general? But this is also a state-funded project. We have a limited amount of money, and we're trying to find an approach that also is very cost-effective. You know, we can't do whole genomes because we're limited on cost. So we're targeting the CO1 region. And we're also trying to generate a huge number of these barcodes. So throughput is also a really important consideration. So we're actively working on developing a workflow that kind of addresses all of these 3 things. So I'm going to start with just touching on effectiveness. And what I mean by this is when we're working with historical specimens, museum specimens, we have highly degraded DNA. So this makes it very, very challenging for us to get these CO1 barcodes. The CO1 region of the mitochondrial genome is the 658 base pair region. And then you add about 40, 50 base pairs of primers. So that's what we need to be able to amplify CO1 and get CO1. Unfortunately, through time, the DNA in specimens becomes highly fragmented. So you can see this distributional graph on the right-hand side. We have on the x-axis, the size of fragments of the genome that they sequenced. The y-axis, you have percentage of those fragments that are of that size. And on this kind of z-axis, you have time. So 2001 all the way to the 1950s. For this study, 2001 was about 20 years old. And what you can see from this is the larger fragments are pretty much gone by 20 years old. There's not these large fragments. And this orange line is where kind of what we need to get the full CO1 region. So for most of these ages, there's no fragments, pretty much nothing that's long enough to amplify CO1. This is another example that was focused on Lepidoptera, where they were actually trying to amplify CO1. So you had percent success on the y-axis and the specimen age on the x-axis. The black line represents the actual 658 base pair region. And you can see pretty quickly, as the specimen gets 10, 15 years, the percent success starts dropping rapidly. The blue and the green line are smaller fragment sizes of CO1, so green being 400 base pairs, blue being 300. So you can see for older specimens, we have more success in getting these smaller fragments, which you can see reflected by the graph on the right as well, that we have some smaller fragments available. But still, as we get older, the success just goes down and down and down. So this is the big problem that has prevented people from utilizing natural history collections to do this kind of bulk generation of a CO1 reference collection. So how can we solve this problem, and how can we actually work with DNA that's so degraded? There's an approach that we can call amplicon tiling, which is this idea that we can amplify and sequence multiple regions of CO1. And then once we sequence them, we can kind of glue them back together like a puzzle. And then we'd be able to get the full CO1 region from these smaller fragments. And so this is what, for the past 10, 15 years, what people have been trying to do with natural history collections. This is one of the paper that was kind of one of the largest efforts to try and do this. They focused on Lepidoptera specifically, but their workflow, they had this huge number of primers that was specific to Lepidoptera. And then they just did PCR after PCR after PCR to try and get something. So this works, and it allows you to take all those different individual fragments and put them together. But it's also not very high. The throughput is very limited by having to do so many different reactions, as well as it's costly to do something like this. So what's been actually used in viral genomics over the past few years is this amplicon tiling approach, but it's paired with what we can call a multiplex PCR. So if you see these two vials, what we can do is amplify a bunch of fragments in two separate PCRs, sequence those, and then glue them back together. And this is something that now we're trying to adopt for actually sequencing CO1, so that we can, in two separate PCRs, instead of going down over and over and over again with different primer sets, try and get all of CO1. So if we imagine it this way, we can sequence potentially 3 to 5 different little fragments, and we can amplify them in two separate PCRs. Once we sequence them, we can glue them back together and have this full barcode region. So this is not something that's being done frequently. There was one paper that attempted this for flies, but that's about it. And so we're really trying to push this forward and develop a novel workflow that could work for our natural history collections. The other benefit of this approach is that even if a lot of those fragments don't amplify, the idea would be that at least we get one. And why that's important is that even these small fragments, maybe 150 base pairs to 300 base pairs, will give us a tag that is efficient at resolving species-like differences, species-like units. So even if we don't get the full CO1 region, by using this approach, we can at least get something that expands our knowledge of insect diversity and gives us a tool that allows folks to start identifying Californian insects. So over the past few months, this has been our focus, is really designing this set of primers that's going to allow us to get at this problem of degraded DNA. So we now have these 2 primer pools that we've designed. The first one is using 2 forward and 3 reverse primers, and that ends up amplifying 5 different amplicons. So you can see that from this gel here, there's 5 different amplicons that are amplified through this. So we've been working with very fresh DNA. I caught these flies and we just extracted them. I've been working with those just to get the multiplex really optimized. So what's great about this is that first PCR, if we get the red line, that's the full barcode. We don't need to do any more work. We have it, we can sequence it. Or if we get these 2 orange lines, those also glue together to form the full CO1 barcode, and we don't need to do anything else. So what that means is that this first PCR we can use across all of our specimens, and for those that we get those regions, we don't need to do anything else, and that's really saving us a lot of time and money if this works for those. For specimens that are more degraded, we have a second PCR using 2 forward primers and 1 reverse primer. And this is what really gives us the ability to tile these amplicons, where this 271 base pair fragment can tile with 2 of the smaller fragments from the first PCR and create a 550 base pair region. That is what we call bold compliant. We can upload it to bold and it'll be assigned to this species-like unit. So through these 2 PCRs, we can actually tile something and get something close to CO1, which is going to give us a lot of power in exploring these and success with these historical specimens. And so what's great about this is all of those amplicons, those little tiny amplicons we're going to glue together, are less than 300 base pairs. So if you look back at this distribution, you can see that at this region, it's a low percentage of what's available, but there are fragments up to 400, 500 base pairs. So we're really hoping that this approach of amplifying these smaller fragments that we can glue back together will allow us to have higher success in sequencing specimens that could be from the 1950s or even older. And the other part of this for effectiveness is trying to do this for a broad taxonomic coverage. So a lot of the papers thus far have been really focused on flies and moths, butterflies, and so they're not really broadly applicable to these collections. And as we were discussing earlier, we really want to get all California diversity. So a part of this is also trying to design multiplex primer pools that will allow us to really amplify across a huge number of insects. So this is how we've been designing primers. It looks very chaotic, but we took 1,400 genomes of insects, each of them from a different genus, and have been using that to perform kind of in silico PCR and understanding primer binding. And we've been able to generate a pool that on average the primers will bind to with 83% of the insect genome. So we're really hoping that this pool we've designed will get us the taxonomic coverage we want. And this is really important for effectiveness when we're working with a collection like we have. So again, this is working towards effectiveness. We're designing this novel workflow that will hopefully be effective for large, diverse arthropod collections. And so through this approach, we will provide this to other people as well. So the idea being that museums throughout the US, throughout the world, will be able to adopt an approach like this that will be both effective, but also efficient in terms of cost and time. And so that we can start actually generating the biodiversity information we want. And this multiplex approach is also, as I kind of mentioned, important for cost because we're decreasing the number of PCRs we need to do. And that's really important. But we've also optimized our approaches in a few different ways as well. We've designed a pretty novel extraction approach that is a lot cheaper than the kits that are normally available. So for working with ancient DNA, there are some available kits, but you're talking about around $6 per specimen, and that's just not realistic for a project of this size. There are some cheaper approaches, 50 cents to $1, which is great. But if we're trying to sequence 100,000 insects, for instance, at maybe $1.50, that portion of that budget can't be just for extraction. So that's an area of cost that we've really worked towards. And the approach we've settled on is a bead-based extraction protocol that actually is just 6 cents per sample. And so this is using spry beads that are normally used for kind of library cleanup for sequencing, but we've built on some other protocols that have existed to create a protocol that allows us to lyse specimens and then clean that lysate up with these spry beads and allow us to isolate DNA and have a really high quality extract that is actually archival quality. We've gotten this cost down so much because we were also making our own reagents. We make our own cell lysis buffer, we get powdered proteinase K and make that, and then we're also making our own beads. And so all of this has really reduced the cost. And so what Julia was mentioning earlier as well with the plating, this gives us also a nice little workflow where depending on the specimen size, we can lyse it for different times. We can use different amounts of proteinase K depending on sclerotization. So this also is a high throughput approach in that we can get highly effective DNA yields from all different types of specimens by this kind of decision tree we've been able to come up with. The other cost-reducing thing we're doing is we're actually going to be working on Oxford nanopore sequencing platforms, specifically the P2 Solo. And what we're going to be able to do is use inline barcodes. So a little bit confusing terminology. We're talking about CO1 barcodes, but we're also talking about these little unique snippets that will add to the primers that will give us ability to demultiplex after we sequence. So we can combine 65 inline barcodes with the Oxford nanopore barcoding kit, and that will allow us to put 6,200 specimens on a single sequencing run. And so that's huge in increasing both our throughput and as well as reducing cost. And so the other really powerful thing about using the P2 Solo, which is one of the cheaper sequencing platforms we can use, one, democratizing this method in general. It's a tool that's more accessible to different labs, but it also allows us to get a huge amount of data per specimen. So there is a lot of other issues with degraded DNA as well as bias that comes out in PCR. And our hope is that by using a platform where we can get really high read depth for every specimen, we'll be able to bioinformatically address some of the challenges that are also associated with these historical specimens. So that really gets us to this cost, effectiveness, as well as throughput. So our extraction protocol is only about 1 hour, 1 to 2 hours of direct labor. The multiplex PCR with these degenerate primers allows us to do very few PCR and get as many these amplicons as possible. And then by sequencing on these platforms, we will be able to do less sequencing runs and pool a lot of specimens onto a single run, which is really, so we're really getting this down of being able to address all these three components. And then I just wanted to highlight a little bit more about why the CCG is a great place to be working on this and the tools we have. We have a lot of space in this lab. We get to have our own lab bench. It's a shared lab space, but we're the largest project. So we have a designated area with our, 2 full-time technicians. They're working every day, very hard, doing PCR after PCR after PCR all the time. So we have a great facility to be working in and some really powerful tools that are helping us process a huge number of specimens. So we have trusty multi-channels and repeater pipettes, but we also have a 96 well benchmart. So this is a semi-automated pipetter that allows us to work in 96 well plates really effectively. So we're using that for DNA extractions, and we're hoping to get a different head that allows us to also do smaller volumes and use this for PCR. And we also have a tool called a tissuelizer. And this is a tool that allows us to homogenize tissue by putting a little bead in each well and shaking it up. That allows us to grind up tissue instead of having to do it manually. So we can process 2 plates at a time in terms of this homogenization. So that's great. And these are just some examples of that. Holly loading a huge gel. We have a big gel rig that we're using to load 4 different plates at one time onto a gel. And then CC using a multi-channel to take lysate off of these plates that we're working in. So just some examples of how we're actually doing this. We're also working with Tosh and the engineering team, this has been so cool for me, having that resource here, to generate some things for the lab rather than having to buy them. And so we've actually laser cut our own combs for our gels. The top one is what we buy, the bottom ones are what we've laser cut. So this is another area where reducing costs as well as helping us increase throughput by creating some of our own tools. This is another example. We have these little plates to put in the tissuelizer, but they didn't actually fit our plates. And so Tosh helped us design a little insert that would allow our plates to fit perfectly in the tissuelizer. So these are just some resources that we're using just to reduce cost, increase throughput, and generate this really effective workflow. So getting to the conclusions of this, we want to develop this workflow, and I wanted just to bring us back to the applications and where we're kind of at. So if we think back to this idea that we can use this data to monitor biodiversity and look at recovery, we want to understand, maybe an example of this, we want to understand, for instance, riparian habitat, how riparian habitat is recovering. We know macroinvertebrates are really fantastic because they're sensitive to environmental conditions, so they're a great group to study how riparian systems are responding to any type of pressure. And so we can use a metabarcoding approach again to take a whole community of macroinvertebrates and sequence them, but we need that reference library to do that. And so right now, our focus is actually using this incredible collection of aquatic insects collected by Larry Serpa, who collected, I think it was 150,000 specimens of aquatic insects. So that's what we're actually targeting right now, so that we can just off the ground right away be starting to generate a database that will be really useful for different projects. So this is currently what we're targeting in terms of all the extractions we've been doing, and all the specimens we've been working on currently are from this collection. And so pairing that together, this large database of CO1 barcodes from this aquatic insect collection, that will give folks the ability to go and do these macroinvertebrate samples in riparian habitat and start understanding how those are responding to any types of pollution or other disturbances. So that's just another example of applications and what we're really currently targeting. Just wanted to transition a little bit to future directions. So we're currently in the CCG still optimizing a lot of our PCR approaches and designing primers. We're really excited to be getting towards our first sequencing run. So hopefully in December or January, we'll be actually getting some barcodes back. And then we're really still continuing to try and develop this really novel workflow that can be adopted by other institutions. So fine tuning as we go. From the collection side, everyone's continuing, of course, very hard to develop the new collections, but there's also now funding to expand to soil arthropods, which is this group that really is super understudied because there's so many small, tiny organisms that are really, really hard to identify. So we'll be expanding to soil arthropods as well. So that's some just cool future directions. And just to wrap it up, bringing us back to the beginning of this and why we started this project to begin with, we are in a biodiversity crisis, and we want to figure out ways that we can start understanding how biodiversity is changing. And arthropods and insects are a huge component of that. And we know from these different studies across the world that this is an important problem. And so by combining these new collections, sampling all across California with the incredible collection we have here and generating this reference library, we are generating something that's incredibly powerful and will inform our approaches in terms of habitat protection and understanding how insect and arthropod diversity is changing as a whole throughout California. So thank you so much for listening. We have a huge amount of people to thank, especially Dan and the California Institute for Biodiversity for their funding, but a bunch of other people. I'm not going to say all of those, but just thank you so much for everyone who's been involved and the Ento team. I'm grateful for y'all from the CCG perspective. But yeah, that's it. Thank you so much for listening, and we're happy to take questions.
Thank you so much for that fantastic talk. We've got a lot of kudos in the chat and a couple questions that have come in online. First are from two of our curators at CAS. The first from Jack. I think Chris and Julie, this might be for you. Would it be possible to collect for you all at some of our study sites? Example like Caples Creek. When they're in the field, can you cross-train our scientists? And the second one from Sarah would be, how are you identifying the ecosystems in which you're collecting?
Thanks. Yeah, that's a great question. Yes, please. We will put you to work for us. So if you're in the field collecting, please bring us anything you want. I mean, we can really take anything from something you find randomly in your field site to we will give you the tools that you need to come back with more like especially leaf litter samples and things like that. Like we'll put you to work, absolutely. And identifying the biomes and the regions that we're targeting is kind of a multifaceted approach because we're looking at what we have in our databases today, which is a fraction of what's actually in the collection, and extrapolating from that saying, okay, these are areas that either have high biodiversity, high endemism, or are not collected very well, so let's go there. And, you know, we've definitely found some amazing sites, you know, outside of Joshua Tree has produced, I mean, dozens of new species already. And then there's the great unknown north, which basically we haven't, like, we have one person, Glenn, up there chugging away, collecting a ton for us, but really we have a lot of work ahead of us to do. So there's a lot. Luis, I see you.
Yeah, I wasn't sure if there was an answer or not. Okay, amazing, thank you. Mind blowing that California has more insects than the planet has vertebrates. I did not know that little factoid there. So along those lines, I think all three of you touched on that. So I have two questions. The first one, the biggest problem seems to be identifying those things. Is there any money in the grant to solve that, to bring in specialists to dig into some of the gnarly groups, the identifications? And the second question is, the reason why the barcode database and GenBank, and they have so many errors in their database, it's not related to the database itself, it's related to who submits. So if we submitted our high quality data to those databases, it would probably increase their quality many-fold. Why, instead of doing that, are we replicating the effort here and creating a whole new database instead of just sending our data and flagging it as public to Barcode or GenBank or whatever database it is?
For the first part of that question, I'll answer and then I'll hand it over to Anna. Basically, no, there is not funding to bring people in to figure out these dark taxa because the cost of doing that is extraordinary. And this project, we only have 2 years of money. And we have an extension of about 9 months to dig into some soil invertebrates, but it's just a tip of the iceberg for what we can do. It's not nearly enough. I mean, we really would need to do like 40 years of this to get a good sense of what we're doing. You know, it should be day-to-day operations in the department, really, but it's just so expensive to do that. So we're going to be generating a lot of work for the future. And I'll hand this over to you.
But you also got a lakeside visitor that's coming from Baja California, right? That would help.
Yeah, we're utilizing, and that, you know, as much as we can, we have these amazing opportunities and funds like Lakeside to bring some people in. So we're looking for those people that we can dovetail into the project as well.
And the other institutions that we're working with as well have specialists in obviously different things than we do. So we're able to kind of collaborate with them still. Even though we're all collecting in different places, we're able to share that knowledge. So having all of the specialists that are working on this project, we can trade and learn more about them.
Yeah, and we're prioritizing what's been already identified, at least for the CCG, for all the collaborators we're working with who are sending us plates. We're asking them to only send us things that have been identified. So that's addressing that a little bit. And sorry, I should have been more clear. We are using Bold, so we're going to upload everything to Bold so that it will go in their bin system and be publicly available. The big thing is just we want all of it to be public and we want all of it to be a resource for other folks. And so we are using those databases, yeah. And that's kind of the point of trying to generate that 550 base pair region at the very least, because that's Bold compliant. They'll put it in one of their bins. That's kind of our target length for that reason.
I'm just curious, and I don't know if you can answer this, but when you were talking about all the database stuff that's up in Canada and like 50% of it isn't available in private, what contributes to that? Like why, how, who, like what's the larger social piece?
I don't actually know, but I think you do.
I think it's because a lot of people are funding their research independently, or if you have a grant to work on your taxa, you can contract BOLD to sequence for you, and then those records are redacted until you publish. And that just proliferates across BOLD, where you have now labs that are getting regular amounts of funding to just crank out sequences, and you're getting 50% of those private now and redacted awaiting publication. So
Such a cool talk, such an amazing project. Thank you so much for the presentation. And like my mind is blown away. So much innovation, and I love to see Tosh engineering solutions for the lab. My question, I'm really excited about using the Larry Sherpa collection to start. I know you're coordinating with other museums. Do they have like a sort of similar ways of prioritizing that are very complementary to what we're doing?
yeah, I think that's a great, thank you for that question. We're very lucky. We're fortunate to have this SERPA collection because it's not only relatively recent, but it's also authoritatively identified by an aquatic entomologist who spent his entire career at the Nature Conservancy collecting these insects, which turns out to also have great DNA. So I think we're luckier than other institutions that don't have a collection like this. There's, now that Anna has optimized these workflows to work really well for legs of older specimens, most of our collections do have these specimens that are 50 years old, and every one of us has different expertises, different areas where we have better collections. So we're all working as a community to try to make sure we're not replicating too much. And just by the nature of our collections, we avoid that pretty easily because we've all had different, curatorial and research expertise over the years. And it's working out really nicely that we have these differences in all the collections.
Okay, we have one more question I think we can sneak in here before the end. We have Diana in chat. We missed you today, Diana. So 2 questions, one for collections. Where's your favorite place to collect in California, and where would you like to collect in the future? And then for lab, what has been the biggest surprise in your work so far?
Oh, my favorite place to collect. I think one of my favorite places to collect was when we were able to collect in the Mojave, just because that's a unique opportunity. The fact that we were able to get the permits to do that. And I also like my herps, so I got to see a bunch of new herps. But we definitely need to cover more of our Northern California area. It's just hard to get to those areas. We don't have nice UC reserves to use. It's going to be a lot of BLM land, which could be more difficult to plan those trips. So we need to get there more. So that's like definitely future directions.
Yeah, in terms of lab, there's been so much over the past few months that we've figured out. I would say, at least for me, the most surprising or exciting is just getting the multiplex to work. That's been something over really the last month that's happened. And so that's been super exciting to start seeing that that's working really well. Some of the other approaches we've been using, we spent a long time trying to figure out how to grind up the legs in a high throughput manner, because it can be really hard to get any DNA from these tiny little insect legs. And so we now are using an approach where we put these things called carbide sharp particles that are just like flecks of metal almost with stainless steel beads. And we're getting really high success with that. And so that's been surprising and exciting to finally figure out that protocol. And that's been another last kind of month. So that's all been really exciting from the lab side.
Well, thank you everyone for joining us online and in person. And if you'd join me in one last round of applause for our barcoding team. Thank you.